Okay, so imagine this. A sudden sound makes you jump or, seeing something specific just brings back this wave of feeling and it sticks with you.
Right.
For the longest time, the idea was that emotions just light up a few specific spots, these feeling centers deep in the brain.
The standard view.
But buckle up because some really groundbreaking new research, this paper in Science, is kind of flipping that whole idea on its head.
It really is.
Turns out emotion isn't just localized. It seems to emerge as this sustained wave spreading right across huge networks in the brain.
Yeah, a brain-wide phenomenon. And here's the really wild part. This dynamic pattern looks remarkably similar in humans and in mice.
That conservation is amazing. So yeah, this deep dive is all about that fascinating paper. They were tackling a huge question, really.
Absolutely. How do those brief sensory things, a sight, a sound, like you said, how do they actually transform into these lasting emotional states that, color our whole experience?
And we're digging right into the main research article, plus some really helpful notes and summaries that kind of break it down.
Yeah, pack it a bit.
Our goal today is basically to get into the core findings, figure out how on earth the researchers managed to map these brain dynamics across different species.
Which was no small feat.
Right. And then explore what this shared brain-wide emotional ways actually means for how we understand feelings. And maybe even find new ways to tackle things like mood disorders.
Exactly. Think about how a particular smell or maybe just a phrase can trigger a memory. And that memory shifts your mood for, minutes, maybe longer.
Yeah, totally.
This research gives us a powerful new lens to see the actual brain mechanics behind that, how an initial trigger creates that sustained feeling inside.
So, okay, why was this so hard to figure out before? I mean, it seems fundamental.
Well, studying how emotions actually emerge from something and then last over time, that's been a real challenge. A lot of the research focused on specific brain areas, like the amygdala, often using animal models, which is valuable, of course.
Sure.
But getting a full brain-wide picture of the dynamics, how the activity actually unfolds and changes over time, and then trying to see if that same pattern exists in humans and, say, mice, that was incredibly difficult.
Yeah, comparing across species like that seems super complex.
Exactly. We knew certain areas were involved, like processing threats, but this idea of emotion being a dynamic process spreading across big networks and finding common rules for that process across species, that was the missing piece.
So the core question they were asking was basically, do our emotional states pop up from sensory stuff using similar dynamic brain processes in different mammals, including us?
Precisely. Could they find a kind of shared blueprint for how feelings are built over time in the brain?
Okay, let's get into how they actually tried to find this blueprint, because the setup itself is pretty ingenious.
It really is.
To get a comparable emotional response, something they can measure reliably in both humans and mice, something, a bit unpleasant, but consistent. They used a simple stimulus.
Yeah, just a little puff of air.
Yeah.
Directed right at the eye, the cornea.
Okay. An air puff?
Simple, but it works.
Yeah.
And the key thing they measured the behavior in both species was eye closure.
Makes sense. You flinch?
Right. But what they noticed was really important. It wasn't just a fast, reflexive blink. There was also the second phase, a slower, sustained eye closure that lasted quite a bit longer.
Ah, okay. 2 parts to the blink.
Exactly. And that sustained closure was the behavior they really focused on. That was their marker.
Why that specific part?
Because that sustained part seemed to reflect the more lasting, affective, or emotional component, not just the reflex. And crucially, they found the sustained behavior had the same key features in both humans and mice.
Okay, what kind of features?
Well, it was clearly aversive. Nobody liked it, mouse or human. It persisted over time, well after the puff itself stopped.
Right, it lingered.
It showed generalization, meaning a similar puff, maybe slightly different, still caused a similar sustained response. And here's a big one. In both species, this sustained emotional behavior was blocked if they gave them ketamine.
Ah, ketamine. Okay, that's interesting. It didn't block the initial blink, just the sustained part.
Exactly, just the sustained affective part. So that shared behavior and the identical response to ketamine gave them this really solid, conserved link to an emotional state, something they could then hunt for in the brain activity itself.
Got it. A reliable behavioral anchor.
Yeah.
So while this air puff is happening and they're watching the eyes close, They're also recording brain activity.
Yes, and across large areas simultaneously in both species, which is where the tech gets really impressive.
How they do that.
In the human participants, who often already had electrodes implanted for clinical reasons, they used intracranial EEG, IEEG.
So recording directly from inside the skull.
Yeah, giving them incredibly precise timing information about electrical activity. In the mice, they used whole brain neuropixels.
Neuropixels.
Yeah.
Those are the super fine probes, right?
Exactly. They can listen in on hundreds of individual neurons firing all at the same time across really wide stretches of the mouse brain. Massive data.
Wow. Okay, so high-resolution stuff in both.
And they didn't stop there. They backed these up with techniques giving a broader spatial view, too. Things like MEG and fMRI in humans.
Measuring magnetic fields or blood flow.
Right. And functional ultrasound, or FUS, in mice, another way to image brain-wide activity.
So a multi-pronged approach.
Oh, yeah.
What did all these recordings show when the air puff happened?
They revealed this remarkable biphasic dynamic, a clear two-part pattern in the brain's response, and it was consistent across both species.
Biphasic. Okay, 2 acts, like you said, with the blinking. What was Act 1 in the brain?
Act 1 was a fast, widespread broadcast of the sensory signal. It was like the news air puff just hit the eye, got instantly blasted out to many, many different brain regions, very quick, very broad coverage.
Okay, initial alert system goes off everywhere.
Pretty much. But then, right after that initial sensory blast, a totally different pattern emerged. Act 2.
What did that look like?
This wasn't just quick processing. It was a slower, persistent activity pattern. And critically, it wasn't confined to just one spot like the amygdala. It was distributed across wide networks, spanning different parts of the brain.
So not localized, but spread out and sticking around longer.
Exactly. This persistent activity just lingered, sort of holding on to the signal long after that initial puff was gone.
And here's where the ketamine comes back in, right?
Yeah.
Linking the brain activity back to the behavior.
Precisely. This is where it gets really cool. Just like ketamine blocked the sustained eye closure, the emotional behavior, it selectively blocked this persistent activity pattern in the brain in both species.
Whoa, okay. So the initial fast broadcast was still there.
Untouched. Ketamine didn't affect that first quick signal, but that slower, lingering neural activity, the persistent part, was dramatically reduced by the drug.
So that's a direct link. The brain pattern that persists seems to be driving the feeling that persists.
That's the strong implication. It's a direct neural correlate for the behavioral effect they saw with ketamine. It really points to this persistent brain activity pattern as the engine driving that sustained emotional state.
Okay, so let's zoom out. If we put all this together, the picture emerging is like a new model for how a quick sensory thing turns into a feeling that lasts.
Yeah, exactly. It's this two-step neural dynamic. First, that quick, broad sensory warning signal,
The broadcast.
followed by the emergence of the slower, persistent activity that's spread across specific networks.
And they had an analogy for this persistent part, right? Something about a piano.
Yeah, fantastic analogy. Think of it like the sustain pedal on a piano.
Okay.
When you press the sustain pedal, the notes keep sounding even after you lift your fingers off the keys, right? The strings keep vibrating.
Right, it holds the sound.
Similarly, this brain-wide persistent activity seems to maintain the neural signal over time. It lets that brief sensory event, the air puff, or maybe a sudden loud noise, or seeing something meaningful, it lets it resonate in the brain and become a more lasting internal state. A feeling.
So the persistence is the feeling taking hold. It creates this, what do they call it? A specifically distributed neural context.
Exactly. It's like this sustained neural atmosphere or backdrop that hangs around, underpinning the emotional state. It helps you remember, oh, that was unpleasant, or influences your next action.
And ketamine's role becomes much clearer in this picture then.
Absolutely. It doesn't stop the initial note, the sensory input. Instead, it seems to speed up the decay rate of that persistent activity. It's like it lifts the sustain pedal way too soon.
Cutting the feeling short.
Yeah, it cuts the neural note short, preventing that sustained emotional state from fully forming or lasting as long as it normally would.
Do they see other effects of ketamine too?
They did. They also found it affected what they call the intrinsic timescale of baseline activity in these networks, basically, the inherent speed at which these brain areas fluctuate. And it reduced the coupling, the communication, between different regions within that persistent network.
Okay, that's a really compelling model. Now, the innovation here, Just pulling this off seems incredible.
Oh, it's off the charts. I mean, first, just the scale on the dual species approach.
Right.
Recording behavior and brain activity together, this comprehensively synchronized across humans and mice, using this whole toolkit, IEG, neuropixels, MEG, fMRI, FUS.
A lot of different techniques.
It's a massive technical and logistical challenge. You're talking multiple research centers, coordinating everything, generating over 100 terabytes of data. It's huge.
Just managing that data sounds daunting.
It really is an Olympic-level effort in neuroscience. But, beyond just the measurements, the computational side was also revolutionary.
It was so.
They developed this new computational tool called Deep SRM.
Deep SRM. Okay, what does that do?
Imagine trying to compare, say, brain activity in a tiny mouse brain with activity in a much larger, differently structured human brain. It's like comparing apples and, well, very different oranges.
Yeah, how do you even line them up?
That's what Deep SRM does. It's a clever mathematical method, a form of machine learning, deep learning, that allowed them to take these vastly different raw brain signals from humans and mice and align them into a kind of shared computational space.
A shared space, like a common language for brain activity.
Kind of, yeah. It finds the underlying shared patterns, the shared dynamics, despite the surface differences. It's a great example of combining the physical measurement with smart mathematical analysis to find those deeper, conserved principles.
That's a huge conceptual leap, isn't it? Finding a shared map for brain dynamics across species like that.
It absolutely is. It's what allowed them to see that conserved pattern, that wave of activity moving from sensory areas towards limbic areas over about 15 seconds after the air puff.
Even though the brains look so different.
Exactly. It's like discovering the same operating system running on completely different computer hardware. It revealed this fundamental conserved wave.
Wow. And they didn't just find correlations, they actually tested if this pattern causes the behavior.
They did. They went for causality using optogenetics in the mice.
Right, using light to control neurons.
Yeah. So they identified a mouse brain region that seems analogous to a human area involved in this persistent network, part of the prefrontal insular cortex.
Huh?
And using light, they essentially replayed the specific activity pattern they'd observed in that region during the sustained emotional response.
So they artificially created the brain pattern.
Exactly. And when they did that, they could actually cause the mice to show that persistent aversive eye closure behavior again, and it lasted for several minutes.
No way. So triggering the pattern triggers the behavior.
It's a strong causal link. It shows this specific distributed pattern isn't just happening alongside the feeling, it seems to be actively driving it.
That's incredibly powerful evidence. So theoretically, the big takeaway here is this brain-wide hierarchical wave model for how emotions emerge.
That's the proposal. It really moves us away from thinking about emotions as just lighting up little isolated spots.
Right.
It paints a picture of emotion as a much more dynamic, distributed process that unfolds across widespread networks over time.
And the practical side sounds just as exciting. You mentioned Deep SRM.
Yeah, it's not just some tool they used internally. They're presenting it as the first public framework designed specifically for comparing neural dynamics related to emotion across species, all on a unified axis.
A shared ruler for emotion dynamics across species.
Basically, yes, which gives us that crucial new coordinate system they talk about.
Why is that coordinate system so important?
Think about developing treatments. If we find, say, a spot in the human brain that seems promising for deep brain stimulation to treat depression, this framework could help researchers pinpoint the directly analogous network dynamics or target in a mouse model. That allows for much more precise preclinical work to figure out how the treatment works, potentially speeding up the development of new therapies.
That translation between species is often a huge hurdle, right?
A massive one. This could be a significant step in bridging that gap. And the fact they made that enormous 100 terabyte data set open access, that's a huge contribution to the whole field. Other researchers can now mine that data for years.
That's fantastic. And you mentioned the timescale modulation aspect before, how ketamine affected the speed or decay. How does that relate to disorders?
Well, the author suggests that problems with the stability or maybe the intrinsic speed, how fast or slow these persistent activity patterns form and decay, might be fundamental to some neuropsychiatric conditions.
Like depression or anxiety.
Exactly. Perhaps in depression, that sustain pedal gets stuck down for negative states, or maybe in anxiety, it's too sensitive, or it could influence why some people respond to treatments like ketamine and others don't.
So maybe the issue isn't just what areas are active, but how they sustain activity over time.
That's the idea. It opens up new ways to think about the underlying mechanisms and potential treatment targets focused on these temporal dynamics.
Okay, so let's try and pull back from this really dense but super impactful deep dive. The big takeaway seems to be our feelings aren't just little blips in isolated brain spots.
Not at all.
They emerge as this conserved, brain-wide wave of persistent activity. It's a process that takes a fleeting input, that air puff, or anything in life, and transforms it into a lasting internal state, a feeling.
Right, the sustained pedal mechanism.
And crucially, this process, this sustained pedal, can be tuned. We saw how ketamine seems to work by speeding up the decay of that emotional wave, lifting the pedal too quickly.
Exactly. It gives us a fundamentally new way to think about the basic building blocks of our feelings.
Yeah. It feels like a framework for understanding how these complex emotions we all navigate every day actually arise from these basic, conserved brain dynamics.
And it's relevant for everyone's daily experience of mood, but also critical for pushing forward our understanding and treatment of conditions like depression and anxiety. It connects the dots in a new way.
So, final thought then. If the brain uses this widespread, persistent activity, this sustained pedal, to hold on to emotional signals and make them last, what other kinds of thinking, what other cognitive processes might be built on similar brain-wide dynamic principles? Could understanding and maybe even learning to tune these neural timescales, could that unlock new therapies or maybe even new capabilities we haven't thought of?
That's a fascinating question to leave people with. If this is a fundamental principle for emotion, where else might it apply? Something to definitely mull over.